Hi student, in this video we're going to talk about the structure of the DNA briefly from what you read in the textbook and then show you how to go from one template strand of DNA to build the complementary strand of the other strand and then go from the template strand of the DNA and transcribe that into RNA. So first starting out, let's talk about the anti-parallel directionality of DNA and structure. As you will see on the left, here is a template strand right here on the left, and the DNA polymerase is trying to build a new strand on the right. So how does the DNA polymerase do that? First, it's going to go from a specific direction. If the template strand is 5' to 3', bottom to top, the new strand will always be synthesized on the opposite end, which is 5' top to 3' down this way. So let's first set it up. So what are we seeing here? We said the template strand is 5' A, C, A, T, C, and then 3'. So what about the complementary strand then? The new strand will always start making in the 5'. So that will be opposite of the 3'. And then if we see a C, like so, we always put down complementary to that, what? A G. And if we see T, we put down complementary to it, an A. If we see A, we put T. If we see C, we put G. A, T, A, T. And now the direction. If this is 5' over here, then the direction on the other end will be 3'. Okay, now that we've gone over that figure, let's try an example. Okay, if we have, say, on the template strand, 5' A, T, T, G, C, C, the other end will be what? 3'. That's right. And let's say this is template strand. Let's try again. How do I find the complementary strand, which we call coding strand? Okay, so what we do in this case is, remember, we always find the opposite. So if it's 3', this must be 5'. If we see C, what do we put? G. That's correct. And then G. And then what? G, we put C. T, we put A. T, we put A. A, we put T. And then if it's 5', then the complementary needs to be the opposite, which is 3'. Now we have finished the coding strand. So this first part shows you how to write out the complementary strand. Now, let's talk about, well, then how do I get the mRNA through the transcription process? So how we do that is first identify which one are we trying to copy. On the exam or in the assignments, identify the template strand. In this case, it's the top strand. Okay, so we're going to try again. If we see 5, we're going to put 3 because it's complementary to that. And if we see A, what do we put? We actually are going to put U. So know that RNA does not speak T. It speaks instead uracil. So when you see A, put U. When you see T, you put A. T, you put A. G, you put C. C, you put G. C, you put G. And then if you see 3', you put 5'. Now you have finished your RNA. So that's a main distinguish between RNA and DNA, is that the nitrogenous base does not include T, but U. So now you have finished writing out the transcript of a DNA template strand.